So I have a couple of confessions. One is that my voice isn't great. That's secondary in nature. But the primary one is I've never been to a Glyco conference. So this is my first Glyco conference I've attended. My students have gone, and I've been working in the Glyco field peripherally. I'm a material scientist. I'm not a glycobiologist, but my students have always gone, and I have incorporated some form of glycobiology in my work. And so I'm nervous because I'm full of glycobiology. I'm going to talk about glycobiology, but nevertheless. Anyway, so here I am. I am a material scientist. I work mainly with biomaterials. I ask biological questions associated with biomaterials. Biomaterials are, by the way, used in medical implants and medical devices, and that's where I'm coming from. I am a director of a National Center for Medical Devices, and we have multiple universities in Ireland which are part of that national center. So from the glyco side of things, I worked with NIBUT. Roshin is around. Roshin was in NIBUT before, and then I also worked with Radka Fahi and Pauline Rudd, and that was my initial interaction, along with Lokesh Joshi, who was active in that space. So that was my introduction 10 years back in the glyco space. So as I said, I'm a biomaterial scientist, and what I'm going to do today actually is I'm going to talk about my own journey in the glyco space, and then give you an example at the end of what exactly we are trying to do. So there will be snippets of small stories, just saying this is what we did and this is what we got, but this is what we're going to do now. So. As I said, I'm a medical implant person, medical device person. I work with biomaterials. So when we started our journey, or at least when I started my journey, I was mainly looking at what is the response of a biomaterial when we implant it. This was 30 years ago. That's what we did, is looking at X, we put a material in, this is inflammation, causes inflammation, chronic inflammation, acute inflammation, what goes on. But we all know, at least we know in our field, that most implants fail. And they are going to fail. And the reason they're going to fail is because we are going to live longer than those implants. Because when implants were designed, they were designed for a particular period of time. So whether it's a hip implant or a heart valve, we live longer, and the implants will not live that long. In fact, they're not living implants anymore; they're dead implants. So you'll need a replacement for that. So that's the context of my work: is looking at can we improve these material systems for better integration in the body. So we looked at drug delivery systems. We incorporated molecules, different biomolecules that we read in the literature, and then we started looking at that space. Didn't necessarily work. We came back to zero and said, well, maybe with looking at different omic responses, transcriptomics and all that stuff, we can get our targets and do it. Not necessarily that worked either. Then we came back to the whole story saying, okay, well, let's look at what is the mechanism of these materials. Are we getting mechanistic responses to the materials that we implant? And that's the stage where the field is at as of now. You can ignore the right-hand side. That's the vanities part of my presentation. But the left-hand side is probably more important, is what we actually do in our lab. So we start with material systems. And we characterize those materials. Even if I'm a material scientist, I work mainly with extracellular matrix-based material systems, and then we look at modifying those material systems by different cross-linking techniques. We look at what is the response to cells at a molecular level, and we can look at level of bioactivities and so forth associated with those biomaterial systems that we work off. Now, this slide should not be, this slide is what our traditional approach in the field has been. So we create building blocks because we can. We as material scientists love to do that. And we love to create structures because we can get beautiful papers. So we create structures just because we can. And then we create linking systems to put therapeutic molecules, and the molecules could be small molecules to cells, to RNAs, nucleic acids, and so forth, and take it to the clinical to a clinical target. This is what I have done in my 30 years of my career, and most people in the field do that. But if I were to start now, I wouldn't do this. I would start from the right-hand side. We've always taken the approach because as engineers and scientists, we can chemistry, we can do structures. Let's put these structures and see what happens. But we've forgotten what the disease host was, and this is where our starting point should be. So again, I think most of you have seen this image, so I'm not going to belabor the point, but this is what was missing in our whole analysis in our field of biomaterials, is looking at the biology itself. So when we look at even drug delivery, we forgot there's a whole huge glycocalyx that a particle has to go through. We forgot that a cargo is lost somewhere. And even if you look at the dimension from a nanoscale point of view, I mean, you're talking about 700 nanometers of glycocalyx, and you're talking nanoparticles. Good luck. You're putting a needle in a haystack. So this is the paradigm that we work with. This field of glycobiology has not really been embraced much by the drug delivery people and so forth. So this is where we need to make the glycobiologist. I would encourage you to go to those meetings and tell them this is what's going on, folks. And the reason one, of course, is from a materials point of view. If you look why we have not embraced it, there's the glycocalyx from a material chemistry point of view is very complex. We don't know what it is. I mean, there's GAGs and heparin, sulfation patterns of heparin. There's O-glycans, N-glycans. We talked about this the whole day today. But this complexity for us is fairly huge from our side of things. So there are obviously gaps, as you know, and I think some of them were alluded to. So we've taken an approach of saying, well, now the tools are there, somewhat there. Can we look at this from our point of view, from the biomaterials point of view, and then look at can we develop therapies associated with that? So in my lab, we have four main clinical targets that we work with: neurodegeneration, Parkinson's, stroke, MS, IVD, which I'll talk about later on, cornea, cardiovascular as well, and then the fundamentally is foreign body response, and this is the area I'm really going to go back to my roots and follow my response after looking at these aspects. So I'm going to give you a snapshot of where our glycobiology piece started off. First, this is Dilip Thomas, who is in Stanford. Now he developed, in my lab, he developed a technique in which we take a material, in this is collagen type 1, we add a cross-linking agent to make it stable, add MSCs, and then we can get a nice microgel-like format. So in this microgel-like format, I'm not going to talk about a lot of data around this, but these microgels are highly angiogenic in nature because they have MSCs and they create a secretome, and this secretome creates angiogenesis. So what we did was we did a large study with 90 animals in a limb ischemia model, took these microgels and implanted into the substrate. We did see fairly nice therapeutic effect, but this is the first time we ever did a mapping of what happens at the tissue level of this vein. This was done with Martina Deschmann's lab about 10 years ago, although the paper was published. It took a long time for us to publish this paper, but what we did see was a high expression of N-glycans in the most angiogenic substrate in the limb ischemia model. So limb ischemia model is the one we developed ourselves in a lab, and then this was the first time we saw, well, from a host response point of view, we see a differential N-glycome seen compared to all the other treatments. So that was the first our entry into the sugar world. Paulo Contesanto, another PhD student of mine, was interested in looking at fibrosis in MI. After MI, you have fibrosis. How do we modulate the fibrosis space? And our point of intervention is not initially when necrosis occurs. It's normally after that, when the inflammatory cascade occurs. That's when we want to intervene so that the scar formation doesn't occur. So we developed our own model. It's a large sheep model where we see MI. So if you see those white patches on the cross-section of the tissue, those are infarcted spots of tissue. So that's where, and we see that, and that functionality, ejection fraction is reduced. So that's the model that we created in our lab. So we have a nice material, which is elastin-like recombiner, which we synthesized. Again, I won't talk too much about this in the interest of time, but by click chemistry, we can get a nice gel-like substrate, which we can inject. Elastin is a predominant component in the cardiac system. We see nice ejection fraction improvement. The blue bars are the hydrogel. We call it the hydrogel material. We see nice ejection fraction, day 28, beautiful results of the sheep, MI being, and the scar formation being reduced. And you can see this by Raman. We also used Raman quite a bit in lab. This is a technique we developed where you can see different patterns. So this is MI only. You see a lot of yellow, which is fibrosis present. You see less yellow in the hydrogel group. That means collagen type 1, type 3 ratio had been reduced, and the healthy itself. Now, if you look at the glyco analysis again, this is changes. This is with Nicholas Carlson's lab in Sweden that we did this work, where we looked at what is the presence of N-glycan. We find that GS1 was highly expressed, and again with high level of macrophage recruitment. And we also looked at the macrophage polarization had been reduced in that context. So again, this is the first time we looked at that, and then we looked at what are the putative structures associated with that, and we see that the alpha-gal was highly expressed at the ischemic area of the heart. So again, this is again an entry into what is the host response in the glycospace. And obviously, I'm not showing you all the functional data that we have, but I'm just looking at the side thing that we did. So then we said, well, okay, let's move from studying the effect. This was all effects. We put this happens. What is the glycosylation response? We were the first in the field to do that, saying we put a material, this is what happens. But now let's reverse this. Now let's look at the host. So we decided which disease target should we use. Obviously, one that is highly full of sugars. So we looked at intervertebral disc. So the approach we said, as I said, was we understand the native tissue first, and then we go and design the material systems, and then let's evaluate that system. So we put the biology first, and that's the paradigm shift that we have moved in our lab. So this is the intervertebral disc. Intervertebral disc is essentially your back, and typically what happens is you go to the doctor when you have pain. When you go to a doctor, you have a pain. That means your disc is protruding on the spinal cord. Intervertebral disc is like a donut-like structure. There's a jelly in it. The jelly is all sugars. It's all GAGs and collagen type 2, type 1, in a much smaller percentage, and it is surrounded by a donut-like structure, which is an annulus fibrosus. So you have annulus fibrosus, NP, which is inside, AF is outside, and that's what we get. So Kieran Joyce, who finished, he's a surgeon clinician who did a PhD, decided to look at what is the profile, what is the N-glyco of this intervertebral disc. So we got human discs in our lab for the first time. Again, these are not very easy to get. These are very, very critical samples. It's not like blood. I saw a lot of people talking about blood. Blood's very easy. Discs are very, very difficult to get because you have to have a traumatic injury to get the disc out. No one's going to donate anybody's disc out, you know. So these are the discs that we got, and this is how, and we grade them in our lab. So we have grade 1 to grade 5. So you can the severity of degeneration is the higher number that you see. So it's grade 1 to grade 5. You can see the gradation of that tissue level. And this is the workflow we did, and this workflow comes from a lot of my collaborators. Richard is one of them, Martina Deschman is another one, and so is Pauline Rudd's group. So that's the workflow we had. I know Anand talked about lectin, but we use lectin only as one of the part of the bigger workflow pace, and we just don't do lectins on its own. So lectin, and then we look at bottom-up proteomics, glycomics. That's the whole workflow that we do on these human tissue samples. So this is the proteomics piece. We saw that ECM proteins are highly expressed, so that's good. So we see that there's a reduction of particular proteins, fibronectin increased in IVD, that's intervertebral disc degeneration. So that's great. That's what we are meant to see, and that's what we saw. Collagen 2A expression, aggrecan expression was also reduced, just like in cartilage degeneration. That's exactly what we see in the disc. What was nice to see that there were two different phenotypes seen. One was a degenerated subphenotype was seen, as you can see over here, and then also the healthy phenotype. So it was good to see that these two nice phenotypes emerge. The first screen, obviously with a lectin microarray, we see that silation is upregulated very nicely. Again, a nice glycophenotype transition between the healthy and the degenerated one, and that was distinctly seen by this. Again, I'm not going to, there's a lot of data over here, so I'm not going to, just briefly what I'll talk about is we did this, it was about a 6-month analysis, or 6 to 8 months. My students spent time in NIBRT doing this analysis, again, manually annotating each single peak. and we find that silation was upregulated. And again, this was done in Richard's lab with gradation of those images, beautiful images of the disc, degenerated disc from grade 1 to grade 5, and this was done by Kieran Joy. So it was, again, a very nice correlation between all of these studies that was done. And we can probe deeper and then say, okay, this hypersilation, so we also did RNA-seq with this, so we can see that hypersilation is setting up this acute response. And this acute response is associated, is also associated and seen in the cartilage field, where it's associated with cellular transferase response to the inflammatory signaling pathway. You can go deeper into this. We also see that glycan branching and elongation was significantly reduced. In other words, nutrition supply was going low, and we can see from the RNA-seq data itself that we can see that the carbohydrate metabolism also had been reduced. So there was some sort of correlation between those two data sets saying, okay, well, the branching is reduced, elongation is reduced, but also all the enzymes associated with metabolism were also reduced. We also did lipidomic analysis in the space, and when we found that the triglycerols and phosphoethanolamine were also upregulated in that space, and also corresponding to that, ferroptosis also was upregulated. So again, we were trying to put all the pieces together, ferroptosis, lipidomics, glycosylation, RNA-seq, all of them, and proteomics together, all of them done on the same tissue sample that we got from the humans. So that's great, that's good. So we got all of these, so many things. Now how do we piece it together? So we did functional analysis. So we take cells, human cells of AFNP, and put the inhibitor, and we inflame them with the inflammatory cytokines. And then we look at inhibitors. So we took at one is fucosylation inhibitor and the silylation inhibitor. And I'm in the interest of time, I won't talk much about the fucosylation inhibitor because that didn't really produce much data. Again, the workflow path, as you can see, was like an RNA-seq, metabolic functional assay, and inhibition assay, and lectin histochemistry. So what we did see was that the inflamed cells upregulated glycosyl transferase. All the enzymes were all upregulated, as you can see in red, and all the blue enzymes were all downregulated. So again, there was a functional point that we saw that these enzymes are also being modulated very nicely, associated with after inhibition. So downstream effect of global silylation also was seen with cell migration and with lectin histochemistry. The fucosylation ones showed toxicity effects. So from a therapeutic point of view, that was a no-go for us. So we can draw a nice pathway over here with looking at mitochondrial function, because this is what actually happens. So we see that there's an altered N-glycosylation effect seen with this metabolics done with the seahorse assay, that we can see that this redox reaction does affect. So ER stress is associated with in the space. So that was great. So just to summarize what we've done, we took human samples, we got our targets. We took an inhibitor. We done functional studies. So how do we translate that to the therapeutic stage? So we are material scientists, as I said before. So we developed a hydrogel, which is a hyaluronic acid-based hydrogel. Put the inhibitor in and did a fairly large canine study. This is about 30 animal study. And this canine beagle dogs are the only species, or the only which resemble clinically relevant model of disc degeneration. That's the only model you can work off. And the limitation there obviously is that we are standing creatures, whereas dogs are supine in nature. There's no other model otherwise you can do, because these are weight-bearing, discs are weight-bearing entities. So again, the same workflow here, lectin histochemistry, glycomic, MALDI, IMS, and we looked at 2 different doses of the inhibitor itself. So I'll just present, there's a lot of data which I'm skipped, but I'll just show, this was done in Martina's lab. This is the imaging piece that we did. This is sham injury hydrogel, which is the HA-based, and then there's a low concentration of the inhibitor and high concentration of inhibitor. This is just the glycan distribution that you see. Now, if you look at, if you do segmentation analysis of that, this is an injury again, and the hydrogel group looks slightly better. The inhibitor group looks much, much nicer, and then the high one is not as great as we see. So again, we see a very nice therapeutic effect that we see from the glycosylation point of view. And you can map that very nicely with silation, and you see that the hypersilation was inhibited in the relevant clinical model that we see in the IVD itself. So what we're doing now, as we speak, this is all unpublished data in the latter part of it, we're looking at functional data points in which has the disk height index changed, which is a functional endpoint, which is what clinicians look for, and we're looking at MRI data associated with. We're obviously looking at RNA sequencing and bottom-up proteomics to see what other effects are happening, and also we are histologically grading the tissue. So this data is coming together. I know it's a very good data that's coming in, but I don't have actual numbers to show that yet. So that's what we are doing. This is how we are approaching things now, is taking biology in our hands, human samples, finding a way to either inhibit, or another way to do that is to actually develop materials which has functionalized on the polymeric backbone that we use. So it's two approaches. One, we can inhibit the phenomena, or maybe we can add epitopes to some other construct that we work in the lab so that they can engage the disease host much better. So where are we going now? We want to complete the IVD. We want to translate that to the clinic. Neurodegeneration, we have completely mapped out the PD as well. We got that published in PNS Nexus last year. It was collaboration with Richard. MS also we have finished. Stroke, we have the samples now. It takes a lot of time to gather all the human samples. So we have tissue samples of stroke patients nicely stratified. Cornea, we have just finished the whole study. Analysis ongoing. I really want to move into the implant host response space because that's where my roots are. So I want to look at what is happening with implants so we can functionalize them and create better implants for the future. I want to couple all of them. In all the studies that we're doing, we are coupling glycomic, metabolomic, lipidomics. Obviously, I didn't write. We take transcriptomics for granted, but those three we want to couple, and also as I talked before, create glycofunctionalized materials as the next avenue that we want to pursue. So a lot of acknowledgements, but I want to thank the patients who donate tissues willingly, and the people who have introduced me to the glyco world: Nicholas Carlson, Richard, who's here, Roshin, Radhika, Pauline, and Martina. Thank you very much.
Thank you very much. So other questions? There was a lot of data and spectacular setups. I didn't completely get how you are actually administrating these inhibitors. So this is in the hyaluronic acid.
It's a gel. It's a hydrogel. So it's injectable. Yeah. So the disc is a.
So it's together, just in the hydrogel.
Yeah, it's within the hydrogel. It's a carrier system. Yeah. It's a hydrogel. Yes. Yeah. So with that, you can control the delivery as well.
And the logic behind having sialic acid inhibitors instead of something
because
else?
There was high upregulation of sialation. And what I should have mentioned before is disc is a very immunoprivileged area. So the off-target effects are not going to be there, as you would see anywhere else, like in cancer or anything else. So it was a conscious decision to choose a target which is immunoprivileged and in a confined area. And it's topical delivery.
Thank you very much. So are there other questions? So if not, then I guess thank you very much. And we move to the next, last topic that you will...
Yeah.